Answer is in itself, anyone. I hope every one of you know phase 1 is the clinical trial where you test your drugs in a healthy volunteers. So if there is already a lot of evidence with respect to that one, as you say, particular major objective of the phase 1 clinical trial is to calculate the MTD. That is the maximum tolerated dose. How much dose the healthy volunteer can tolerate. So if the molecule is already used from the ancient literature, so what is the point to do the phase 1 clinical trial? And if you go through the do the cost analysis of phase 1 clinical trial, you will find that it's very costly. Because first time you are going to test your molecule in a human volunteers, if we talk about new chemical entities, it's just like a ICU setup. So it's very costly process, phase 1 clinical trial. So point is, if there is already ancient literature is mentions about the molecule, so in that case you can skip the phase 1 clinical trial. But of course animal toxicity studies are required. We always assume that herbals are safe, but there are many examples where we found that herbals are associated with various adverse drug reaction as well. So that's why there is a program, Herbal Vigilance, has been initiated by the IUs. I hope you all might heard about pharmacovigilance. That is a program which is under the IPC, and we are talking about the adverse effect of medicinal drugs. With respect to herbals, the program is initiated by the IUs, and that program we call Herbal Vigilance. Right, so what I means there are many case reports are coming from the market which indicate that herbals are also associated with adverse drug reaction. So that's why animal toxicological studies you have to do, and you can see in category 2 there is a new indications or new formulations you are preparing. Suppose you are making the nano formulations of curcumin or piperine or any berberine or any herbal molecule. So in that case, you have to do the phase 1 clinical trial. You can't skip the clinical phase 1 clinical trial because it will considered as a new drug. So I hope every one of you knows about phytopharmaceutical. It's included now in the NDCT 2019 of India, as I am talking about. Earlier, we are following the schedule by if you want to do any clinical trial. So what are the requirements? It's mentions in the document. That document is schedule by. But this schedule by is replaced with NDCT 2019, and that is we call New Drug Clinical Trial of India. So in that, if you go through the NDCT 2019 document, you will find this phytopharmaceuticals is included. So phytopharmaceutical is a drug of purified and standardized fraction assessed qualitatively and quantitatively with minimum of 4 bioactive compounds. So that is a phytopharmaceutical. If you have prepared any extract, so you have to be so there are at least four bioactive components are there. So that will be qualified as a phytopharmaceuticals, and you can conduct a clinical trial and you can put your phytopharmaceuticals into the market with the regulations. Next slide, please. So data to be submitted to the by the applicant if you want to market a phytopharmaceutical product. So what data you have to be submit to the regulatory agency, and regulatory agency in this case, because this is a phytopharmaceutical and mentions by the CDSCO. So you have to submit your application to the CDSCO. I hope every one of you knows about CDSCO. So that is your regulatory agency, which will assess your protocol, which will assess your data which you have submitted and make a conclusion out of it, whether you will get the market approval or not. Right? So in NDCT you will find there are different forms like CT01, CT02, CT03 like that. For different purposes, you have to fill that form. Along with that form, these all data you have to mention with respect to if you want to put phytopharmaceutical into the market. So you can see how much data you have to be generate and you have to be submit to the regulatory agency. That application we call NDA applications to get a market approval. So all these data, identification, authentication, source of plant used for the extraction process, what you have used, if you have prepared the formulation, so what formulations you have prepared out of it, what is the stability profile, what is the safety, is there any human studies are available, so you have to be provide all these data. If you want to learn much more about these things, you can go through the CDSCO websites. So you can see there is a NDCT, and nowadays we are submitting our application through online platform only. They are not accepting any hard copy document. So there is a Sugam portal of CDSCO. In that one you can register You do the login, and after login, you select the city particular form with respect to phytopharmaceuticals, and submit all these data to the regulatory authority. So they will assess it, and depending upon your studies, whichever you have conducted, depending upon data, you can get the approval of your phytopharmaceuticals into the market. So now I just give you the glimpse about herbal medicines, how these and what is the Indian regulations. Again, questions always arise in mind. If there is regulation is also there, herbals are effective as well. There are means clinicians who are using these herbal medicines, Ayurveda Charya, and they are getting the effect. So where we are lacking? What are the challenges with respect to herbal medicines? Go to next slide, please. So challenges with respect to clinical trials. If you go through the clinicaltrial.gov or CTRI, that is the Clinical Trial Registry of India, if you want to search clinical trial, where the clinical trial on what drug it's conducted in India, so you have to be search on CTRI. CTRI is Clinical Trial Register of India, where you have to register each and every trial which you are conducting in the India. Globally, we are searching in the clinicaltrial.gov platform. So if you go through that one, you will find that there are a lot of clinical trial has been conducted with respect to new chemical entities. But with respect to herbals, there are very less clinical trials which has been conducted. And if you go to find the reason why it is so, you will find that Ayurveda Charya or these herbal peoples are not ready to do the clinical trial. They are saying there is no need of clinical trial. But clinical trial is required. If you want to get acceptance globally about your product, they will ask you about the scientific evidence. They will not believe you. If you are saying this is a very good, this Brahmi is very good for the this brain tonic, so they will ask you where is the scientific evidence. Tell me the scientific evidence. Otherwise, I will not accept it. So we have to conduct these clinical trial, and we have to be accept that if you want to get the acceptance globally, so we have to be generate scientific evidence with respect to our herbal medicines. So that will help in a great way. Then on the global platform, your data will be accepted, your herbal medicines will be accepted. So that is one of the challenges. Another challenge is with respect to investigational product. To do a clinical trial, you need an investigational product, and your investigational product should be uniform throughout all the centers. I hope everyone of you know phase 3 clinical trial particularly is a multi-centric trial. We have to do our trial in a different centers. So in these centers, if you are doing a trial here in the Meerut, one same trial you are going to do in the Bombay and in Maharashtra, in Jammu. So your product should be uniform at all the centers. But uniformity of herbal medicine is very difficult to maintain. That is one of the challenge. Another challenge is with respect to standardizations. I think every one of you know standardization of herbal medicines is a very difficult task. You have to be standardized. Means herbal constituents are differ in a different seasons. Same plant, if you are growing it in a summers or you are doing the extractions in a winter, you will get different constituents. There are different locations, differences are there, right? So that is the standardization is also the one of the challenge, and of course stabilities. Your product should be stable, then only you can do the clinical trial, right? Another challenge with respect to herbal medicine is lack of specific research methodology. With respect to new chemical entities, your research methodology is defined. What will be the sample size? With respect to herbal trials, statisticians are not able to answer how much subjects are required to do this trial. Because there is no set of guidelines, there is no set of principles how to do these trials of herbal medicines. There is another challenge is, I hope maybe you have heard about the blinding. Blinding we have to do in the clinical trial to reduce the biasness. That investigator as well as the participant as well as the sponsor should not know about which subjects are getting treatment and which are on placebo. But with respect to herbal medicines, to maintain the blinding is very difficult task. The reason is, anyone? Why we are not able to, it's very difficult task to maintain the blinding procedures with respect to herbal medicines. And blinding you have to do in the clinical trial to reduce the biasness. Otherwise, if I knows what you are getting, what he is getting, so I can manipulate the results. I knows, okay, इसको मैं placebo दे रहा हूँ, इसको investigation दे रहा हूँ, तो there should be decrease in blood glucose, and I am claiming that my herbal medicine have a hypoglycemic effect. It's a human tendency to do the manipulation. So to reduce that, you have to do the blinding. That your investigator don't know which this person is getting, which this person is getting, which that person is getting. Whether this person is on placebo or investigational drug. But with respect to herbal medicine, it's very difficult. Reason is because herbal medicines you can identify by their organoleptic characteristics. By smelling you can identify. The experts who are working in the herbal medicines, by smelling they will say that this is the product I am getting and you are getting placebo. So it's very difficult to maintain, create a placebo as that of the herbal medicine. With as that of the organoleptal. Color is there, okay, color you can do, you can create the same color, but what you will do with the organoleptic characteristics? So that is also one of the challenges which we are facing when we are planning to do the clinical trials of the herbal medicines. Another challenge is with respect to poor pharmacokinetic and dynamic correlation. So I think many of you knows curcumin, there is a lot of literature is available. But still we are not sure about curcumin is going to absorb or reach to the systemic circulations or not. Hello? Curcumin, whatever testing we are doing, we are showing it's anti-inflammatory, this, that. But if your molecule is not reaching into the systemic circulations, or we are not able to detect how much curcumin is reaching to the systemic circulations. Even we have a LCMS or other powerful analytical techniques. So without pharmacokinetics, how you will correlate the pharmacodynamics? That is also the one of the challenge. That's why when we are looking for a research protocols with respect to herbal medicine, some are putting herbal, we want to give 20 mg per kg extract, then they are keeping second dose 30 mg or 40 mg. So I always question them, why you are keeping so close doses in animal grouping? Because your product is herbal. With respect to new chemical entities, you can go through that. But same kind of things you will get at 20 mg, same things you will get in 20 to 40 mg. Unless until if you are claiming that your herbal extract contains very potent phytoconstituents. So dose, because absorption is very much important with respect to herbal medicine. Many herbal medicines have very poor pharmacokinetic profile. So when you are planning to work on herbal medicine, just look for what phytoconstituents you have and what is their pharmacokinetic profile before planning the pharmacodynamics. Because if it is not reaching to the systemic circulation, so what is the point to do the pharmacodynamics, right? So that is one of the major issue with respect to herbal medicine. That's why you will see that we are giving herbal medicine at higher doses. But even at the higher doses, there is a limitations of toxicities as well. Then another one is pharmacovigilance system. I already discussed about all the herbal medicines are not safe. It's a myth. There are many cases which have shown that there is herbal medicines are associated with adverse drug reactions. But causality assessment which we are doing in the pharmacovigilance, it's also the one of the biggest challenge to do with respect to herbal medicine. The reason is you have taken any extract. Extract contains number of phytoconstituents, 100 phytoconstituents. So how you will assure that which phytoconstituent has produced the adverse drug reaction? Because causality assessment means you are going to verify whether this product is responsible for that particular ADR or not responsible. So that you have to be, that is also one of the challenge. And of course regulatory control. Now year by year regulatory bodies are evolving and they are considering the herbal medicines. They have included the phytopharmaceuticals concept in NDCTs, but earlier there is no regulations, fixed regulations for that. So now again scientific evidence, as I said, when we talk globally, outside India they are not accepting our herbal medicine. They ask you, show me the scientific evidence. What evidence you have? And in China, the first drug of choice is TCM, that is a traditional Chinese medicine. And if you see the traditional Chinese medicines and herbal medicine, they are somewhat similar, what I have found. But they are generating a lot of scientific evidence with respect to TCM. Next slide please. So now with respect to how network pharmacology can help to address one of the challenge, that is the scientific evidence. So I am going to address that part, how your network pharmacological approaches can help to generate scientific evidence with respect to our herbal medicine. Next slide please. So I hope network pharmacology, in simple terms, if you want to understand, network means what? Network means if someone have a lot of connections, so that means it has that person have a good connections with others. So like that there is a network, and in biological systems you all know everything is interconnected with each others. Every signaling pathways, targets are interconnected with each other. They are not in isolated form. So pharmacology, everyone understands, studies the effects of the drugs. It includes your both pharmacokinetics and dynamics. So complex diseases, but as I said, there are a lot of interactions among the targets, among at the gene level, at the protein levels. There is a lot of interactions is happen. So network pharmacology will consider all these interactions, and with respect to herbal medicines, you can identify the possible mechanism of that herbal medicines in that particular disease. Like if you are claiming that Withania somnifera is good anti-inflammatory properties, so your network pharmacology will tell you about what is the possible mechanism of Withania somnifera in the treatment of inflammatory disorders. So that is the network pharmacology. Go to next slide. So disease network and network pharmacology, as I said, to understand the drug pharmacological mechanism in network perspective. So it's a emerging discipline useful in drug discovery, which combines genomic technologies, system biology, chemistry through computational biological tool. So new frontier of systematic drug research in the era of AI, and globally network pharmacology is regarded as next generation's medicines research mode. Go to next slide. So I think that is every one of you knows how the drugs showing their actions. So first you have to understand before doing the network pharmacology. I have seen many students are very much motivated to do the network pharmacology, but if you ask them what is the signaling pathway of in this particular disease, you are unable to answer. So how you will do the network and how you analyze the network without the basic knowledge of the signaling pathway. So to do the network pharmacology in a good way, what is required? Again, I always strongly believe that there is no substitute for the basic knowledge. You have to work on your basic. What are the basic? What are the drugs? What are the how the drugs showing their actions? What are the different types of receptors? And when the drug is acting on GPCR, what are the different signaling pathways? What is IP3? Where it's going to act? What is it going to do on DNA? So all things you have to be revise. Then you can think for the network pharmacology. So this is just a drug, how drugs showing their actions. That is it's a visible, but drug effect will be drug effect is visible and drug action is invisible behind that, and that invisible part we are identifying through the network pharmacology. Next slide please. So this is, I think everyone pharmacy students know most of the drugs showing their response by acting on receptors, and there are 4 types of receptors. These are the signaling molecules, these are the primary messenger, secondary messenger, and so on. Next slide please. I am not going in detail. This is also the one of the GPCR pathways where you can see how CMP is increased, how IP3 DAG pathway works. There is a storehouse of sarcoplasmic reticulum is a storehouse of calcium. Then IP3 act on it, and calcium release will act on actin and myosin filament, and so the contractions. Next slide please. So few signaling pathways. Again, what I would like to say is, before doing the network pharmacology, just identify your area where everyone have a different research area. Someone is working on cancer, someone is diabetes, someone on other, and so on and so on. So with respect to areas, please understand in detail about the signaling pathway before planning to do the network pharmacology. It's very important because network pharmacology, how you are doing, you are using some softwares, using some databases, but if you are not able to do the interpretations, so it's of no use. You will interpret in a wrong way as well. So like that, these signaling pathway we have explored, and then you can combine your network pharmacological results with experimental approaches as well. You can plan for animal experimental studies where you can verify it, and you can go for a good publications as well, and you will get a confidence on it. So these are the few signaling pathway which we have explored. That is apoptogenic signaling pathway, which I have done on primary animal cell cultures. Particularly thymus and spleen with the delta-methrin. Delta-methrin is one of the pyrethroid, which these pathway we have explored experimentally first by network pharmacology, and then we have explored it experimentally. Go to next slide. This is another one which we have explored. This is the apoptogenic signaling pathways, how they will work. So I am not going in much more detail, otherwise you all will sleep. Next slide, please. This is another signaling, immune and nervous signaling is something which you have to be, every system is connected with that. Without that, how you will do the network pharmacology? If you are not able to understand what is the toll-like receptor, which toll-like receptor is sensing which part, and after sensing how it is going to activate with signaling pathways, and after activation of signaling pathway, what it is going to do at the DNA levels, at gene levels, then what it is going to do the protein levels. So you can't do the interpretations, because immune system is something which is interconnected with each and every organ. So you have to know, if your innate immune system is not able to handle the pathogens or any exogenous substances, so how these antigenic peptide is hand over to the adaptive immune system. There is a particularly handover system is there. There is antigen presenting cells. There is a MHC, major histocompatibility complex. So there is a complete mechanism by which your antigen is presented to the adaptive immune system, particularly the T cells, helper T cells, regulatory T cells, or cytotoxic T cells. And after that, there is a go under the process of transcription, translation, and do the clonal expansion. Go to next slide, please. So importance of network pharmacology is you can use if you want to identify the potential drug candidate. If you have synthesized any new chemical entity in your lab, now you don't know. You have planned it for anti-cancer activities or any other activities, so you can apply the network pharmacology at this step to identify what are the possible target which your new chemical can hit in that particular disease. That is the one way where you can use, medical chemistry professionals can use network pharmacology in their research. That is the one way. Another way is if you want to understand the drug mechanism, again, whether it is a herbal molecule, Ayurveda, new chemical entity, whatever it is, if you want to understand its mechanism, so you can use network pharmacology. Understanding the possible drug-drug interactions, also you can do two drugs with same target, how it's interacting, what targets you are getting. So we can understand the drug-drug interaction as well, and you can identify the novel drug-drug interactions as well. Then identifying the possible mechanism of adverse drug reactions, that also our lab is doing. We are particularly identifying the novel signal of the drugs. Novel signal means which is not earlier known. So that we are doing the analysis by using the data mining algorithms. And data we are extracting from the FAIRS database, Udra Vigilance, and all these databases, and applying these. And after that we are applying network pharmacological approaches, because it's a new thing which you have identified. So you don't know the, but where means how it produced that effect, what is the mechanism behind it. So that also we are using in that particular area. Next slide, please. So to understand the network, first you have to understand what is the simple network. So there is a, you can see in this slide, there is a circles, there is a line which connecting the circles. So circles, so-called node. These circles in any network, you will see it's a node. So these circles is node, and it can be gene, it can be protein, it can be metabolite. It depending upon on which network you are working on. Some are working on gene network, some are on protein, some are on metabolite networks. Then there is a line connecting these nodes, these we call the edges. The line connecting these circles, this is the edges in terms of network pharmacology. Next slide, please. So important terms related to network is there is a, you will find these terms if you go through the research article with respect to network pharmacology. Someone will write about the hub, this is the hub, this is the degree, this is the betweenness, this is the shortest path. So what are these terms? So you can see the hub is a node with the high degree. And what is the degree? Degree is the number of edges connected to the node. Betweenness, the number of shortest path that go through a given specific node. And what is the shortest path? A minimum path between any two nodes in a network. So these are the parameters which you have to be analyzed after creation of network by using the software, because nowadays we have a user-friendly software. Whatever you will add it, you will get the network. But the important thing is how you have generated the network, what are the parameters. Sometimes what happens, whatever the node we are feeling that it's very important, it's not important that much. So you have to be discussed with the experts as well. Next slide, please. So you by this figures you can understand so much better what is the node and what is the how, what is the degree, what is the shortest path, and what I am talking about betweenness. Next slide, please. So a biological network at steady state, it's also similar things, means why network pharmacology is popular year by year. So these are the proposed based on the theory of network biology and biological balance. So provide ideas for the drug discoveries, because everything you can't test experimentally. You can't do the Western blot for each and every protein. You can't do the RT-PCR for each and every gene. So network pharmacology will refine and help you. Next slide, please. So types of network, as I said, some are working on gene regulation network, some are working on protein, some are working on signal transduction network, some are working on your metabolic network. Next slide, please. Common databases and tool which we are using in the network pharmacology. So now the steps. If any one of you planning to do a network pharmacology, I am telling you the standardized procedure how you can do, and each one of you can do it. Just go through your research area, just go through your signaling pathway. After that, I am telling you how to do it. How you can do by using your laptop. There is nothing is required to do such kind of project. So you have to use common databases, and whatever databases you can see on this slide, these databases are freely available. Everyone can access these databases from your laptop. So first step in network pharmacology is you have to identify the target of the compounds. So if you are working with respect to herbal medicines, so as I said, herbal medicines, if you are making one extract, so that extract can contain number of phytoconstituents, maybe 50 phytoconstituents or 80 phytoconstituents. So first make the list of these phytoconstituents. So these phytoconstituents list you have to be make and draw the structure of these phytoconstituents. That is the first. Then after drawing the structures, or you can get the smiley notation of these compounds, one by one you can search in these databases. That is the first step. Identification of targets of compound. So like identification of target of curcumin, bithanolide, or so many phytoconstituents if you are planning to work on phyto. So first step is Swiss target predictor. You just type in a Google Swiss target predictor, go through its web page. It's very easy to use. But again, important thing is in that database everything is mentioned. First read about database. What is the basis of this database before using the database? Because using of these databases are very easy. What you have to do? You have to just draw the structure or just put the smiley notation of the compound in the search items. Just click on predict target, it will give you the results. That is of no point. You have to first read about the database, what is the cutoff value, what is the p-value, what you have to be keep and what you have to discard with logic. So that you have to go through each and every database. So that is the Swiss target, C database, Stitch database, drug binding database. Up to this, these are the databases freely available to identify the target of compounds. So if you have a new chemical entity, in that case you have a single compound only. So you can use these all. So now question might be arise in your mind, why we are searching same thing in these 4 databases, why not only one database? And in literature you will find there are many articles which are published on network pharmacology, they have used only the Swiss target predictor. But that is not the best way to do that. The reason is every database has their own advantages and own limitations. Like some databases has been updated in 2010 because these are freely databases. They are not updated on daily basis. So you have to use all the databases and make one comprehensive Excel file. And in that case, obviously you can get the duplicates. So you can, by using the Excels, you can just clicking on, selecting on that one and click on the select duplicate, you will get the duplicate, right? So remove that duplicate, but note down the numbers, because when you are writing a manuscript, you have to mention that how many duplicate you have identified, how many you have removed. So each and everything you have to be maintained. And apart from these databases, you have to search or do the data mining of the literature as well. The reason why data mining of literature is also important to do, again, because you are using a freely available database. If you are using commercial one, no one will questions on that, because commercial one is updated on daily basis. But if you are using freely one, academia, we have a limitations, we can't go for a commercial one. So use the freely one. So in along with that, you do the data mining of literature as well. That is the first step. Next step is target involved in disease. So first you have make one comprehensive Excel file where you have put in all the targets which can be hit by the compounds. That is the one file. Another file you have to create targets involved in that particular disease. If you are going to work on cancer, so you have to be put in these databases. DigiNet is the most popular databases to identify the targets which are involved in disease. So you can use DigiNet. DigiNet Plus is a commercial one. If you have a money, you can purchase that. Similarly, you have to search in the target which are involved in that particular disease. For example, you are planning to work on cancer, so you have to put all the targets which can involve in the cancer. Again, you can get the duplicate, just remove the duplicate and make the one comprehensive file. Next slide, please. Next step is, after doing this exercise, what is the next step? Next, you have to identify the common targets. So common target, how you can identify? There is a Vani tool. Earlier we are doing the VLOOK. I hope many of you knows VLOOK formulas in the advanced Excel, by which we can do lot of things. But now we have another user-friendly tool, Vani tool. So Vani tool is also freely available. In Vani tool, just you have to add in one side the target which can be hit by your compound, and in another side the target which are involved in disease. So this Vani tool will give you the common targets. Common target which your compound can hit, and target which are involved in that disease. Because what is your objective? Your objective is to find out the possible mechanism of that particular compound in that particular disease. So you have to identify the common target, right? So that we are doing with this Vani tool. And next is string. So after that, these targets are not in isolated form, they are interacting with each other. So how you will get the interactions among the targets? And targets can be your protein, target can be your metabolite, target can be gene. So there is a tool, string. String is widely you will find in the network pharmacological papers, by which pupils have checked the interactions among the targets by using the string database. There is another tool, DIP, Stitch and Enact. So it will give you the interactions among the common target. In that step, you have to take only the common target, right? Then next step is Cytoscape. Cytoscape you will find every papers, and it's freely available. You can download it. It's not a big software. It's a visualization tool in which you have to add this file, and you can create a beautiful network which you might have seen. Some are presenting with circles, some are presenting with others. These are different ways for the presentations. So Cytoscape, and then you have to do the SR plot, because up to this process you have identified the targets. And these targets will make particular pathway. Like I have shown you apoptogenic signaling pathway. So in apoptogenic signaling pathway, there is a ROS, there is in mitochondria, there is a BCL2 protein, that is the anti-apoptotic. There is a BAX protein which induce the apoptosis. There is a caspase 3, there is a MMP9s and so on. So all these targets makes one pathway, and that pathway is apoptogenic signaling pathway. So how you will get that pathway information? You can do the SR plot. So by that one you can identify that this drug is acting in this particular disease through this pathway, and this is again the prediction. So you have to be mentions maybe, right? You can't sure about it. Experimentally you have to verify it. Next slide please. I hope I am on time. So steps already next. So one by one I have added. This is the first step, identification of compounds or new chemical entity target. Next step. So these are the these are the screenshot of the web pages for where you can use these databases. This very user friendly, as I said, Swiss Target Predictor, C database and Stitch. Next slide. So this is a Swiss Target Predictor. So as I said, whatever the database you are going to use, please read about it. There are a lot of material is available. They have published their paper as well, how this database has been created. If you don't know about the basis of the database, how you will use it in a proper way? And today's problem is this: students are just writing the steps, and they are not reading about the database. They are just putting the structure of the molecule and getting the result. No, first go through the database like Swiss, where it is available, what is its structure predictability, how it predicts the target, what is the 2D similarity, what is the fingerprint vectors, how it's calculated and how it's providing the result. If you are knowing each and every detail, that will give you the confidence and you can speak at any stage, right? Next slide. Without doing that, read about the database. I am not going in detail due to time limitations, but what I am showing that in database there are a lot of information. Read about it, how these predictions has been done by this database, what is the basis for creation of this database, what is the p value, what is the FEP, how it's machine generated or how it's doing the prediction, when it's updated. So everything you have to read about the database before using it. Next slide please. So this is the web page. Now anyone can use the database. It's not a big deal. Big deal is you have to use it in a proper way. यूज करने में क्या करना है इसको, आप ये खोल दिए स्विच टारगेट, व्हाट यू हैव टू डू, यू कैन ऐड द स्माइली नोटेशन ऑफ द कंपाउंड. स्माइली नोटेशन you can get from the PubChem or from the Campbell or any other database. Or if you are not comfortable with the smiley notation, just draw the structure of the compound this side. And what you have to do, you have to just click predict target. इतना सा काम है, आपको क्लिक करना है predict target पे, it will give you the results. Next slide. But you have to see like switch target predictor. If you suppose you are planning to connect your finding with the you are planning to do the experimental studies in rabbit. So you have to see this database is providing the data of which species. So you can see this select species, Homo sapien, Mus musculus and Rattus norvegicus. Only these 3 species data this database is providing. और आपने experimental study कर ली rabbit पे या guinea pig पे, जिसका तो टारगेट ही नहीं निकले थे. How you will correlate it? And when the expert will ask the questions, you will unable to answer that, right? So that means you have not read about the database when you extract the data. Next slide, please. So it's very easy results you will get. There is a, you can see there is a export result. You can export the result in the Excel file, in PDF, whatever you are comfortable. So most comfortable, I think we are with the data when we are getting in the Excel. So you can export the result. Next slide please. So you can get such kind of Excel, say all these. You can see there is a target name, there is a common name, there is a UniProt ID, Campbell ID, these are probabilities and known activities. So you can you can provide the cutoff as well, cutoff value as well, probability as well. Next slide please. Next database is C database, and it's provided by a laboratory at the Department of Pharmaceutical Chemistry at the University of California, and it's a free of charge. But if you go through this database, they have mentioned one disclaimer. You use this database at your own risk. Suppose you have any new chemical entities you have synthesized and you have not patented yet, and you are using these databases, so they have already mentioned you have to use it at your own risk. Right, so they are not giving you guarantee, because online whatever we are submitting, it's not possible that it's not maintained anywhere. It's going to maintain somewhere else, right? So it's free of charge, but it's on risk. So no guarantee about the data confidentiality on this public service website. Next slide, please. So this is the web page of similarity assemble approach. That is a C database. Again, you can see how easily you can use it. You just add the smiley notation of the compounds and just click on try C. Next slide please. So this is another database, this database, and this database contains the data from the genomic context. If you are planning to correlate this data at the gene level, so this database is very important. And it's a freely as well as commercially available, and combined score they are computing by integrating the probability from the various different types of evidences. Next slide please. This is the web page of Stitch. Again, it's also very user friendly software to do. You can just add the smiley notations and click on the search. Next slide. This is another database, drug binding database. This is the School of Pharmacy and Pharmaceutical Sciences at University of California. And it's a public web accessible database of major binding affinities, where you can also identify the possible targets of your compounds. Next slide. This is a web page. So you can see here, I think we have written the routine. So you can click on go, you will get the possible target of the routine. I am not talking about the routine in cancer. That you have to do. These databases will give you the possible target of routine. Next slide please. Next step is identification of disease targets. So I have shown you where you can identify the targets of the disease. Next slide. This is the DigiNet. DigiNet is most commonly used. So there is a DigiNet Plus. If you have a money, you can purchase it. I didn't remember how much costly it is. For commercial purposes, you can use it. If you are using commercial version, so in that case, you can skip the data mining of the literature part. But if you are using freely one, don't skip. You have to use all the available databases and make one comprehensive file. The reason is you don't want to skip anything. You want to include each and everything. So how you can include? If suppose in Nature, recently one paper in 2024 has been published with respect to your compound and they have identified this is a particular novel target and you have missed in your network. Because that database may or may not included your that target, right? So that is you have to do. Next slide. This is the DigiNet web page where you can use it. You can just sign up. It's freely available and do the login. Next slide, please. You can create login ID and password as I have created. This is my login ID and password to use this database. Next slide. And here you can add in the search disease name and you can click on summary of disease association. You will get the target which are involved in that particular disease. So if you are planning to work on cancer, just type the cancer and get the target in the Excel file which are involved in the cancer. Next slide. So like that you will get, like we have extracted the target which are involved in the drug resistant epilepsy. Next slide. There is another gene card. This is established in 1997 by Crohn Human Genome Center of Institute in Israel. And from this you will get the relevance score and download the targets from gene card as well. Many paper, as I said, you will find that researchers have just downloaded the target from DigiNet only. That is not the right way. You can miss many targets. So just download from gene card. Next slide, please. This is the gene card web page where in the search item, again it's very easy to use. Next slide. This is the man-OMIM. This is also very important database if you are working on hereditary diseases. So in that case, this database you can't miss out. So you can get the Mendelian trait disorders and this Mendelian inheritance in man, MIM. And online version was created in 1985. Next slide. This is the man-OMIM web page. Next slide. Online tool for. Then there is a HPO. HPO is a human phenotypic ontology database. And it contains particularly the clinical data and NIH supported project that integrate it. And it's available on this web link. Next page. So this is a human phenotypic ontology where you can get this web page of that. Next. Then another one is Malacard. So this is also Malacard. You can get the target which are involved in the disease. Next slide. This is the Malacard web page where you can get. Again, I am again and again repeating, you have to extract the data from all the available databases. Maybe apart from these there are, and maybe you will get other databases as well. Include that one also. Because when you are making the comprehensive file, in that one you can detect the duplicate and remove it. That's fine. That's not a very difficult step. Next. So data mining approaches. Next, data mining approaches of the literature is very important, you know. You can't want to miss any target which is involved in disease, any target which your compound can hit. So how you can do the data mining literature, maybe many of you are already doing. You can do it by the Embase. Embase is paid one, but Medline is your just PubMed. So PubMed is most popular tool by which you can do the data mining and clinical trial websites as well. Next slide. So how to do the data mining? I have included just a few slides. So first you have to identify your mesh terms. Mesh is the medical subject heading. Use the mesh terms with the Boolean operators. And what are the Boolean operators? Boolean operators are just or and. Right? Go to next slide. So you have to be very careful when you are combining mesh terms with the Boolean operator and or. By changing and or, your literatures will increase significantly or decrease significantly. Next slide, please. So suppose, for example, you want to know the target of rutin in drug resistance epilepsy. So how to do it? Just go to the PubMed and just click on the advanced options of the PubMed. The next slide. So now you can see first one, if you are adding rutin or DRE, then what will happen? If you are using this mesh terms with this Boolean operator and your objective is you want to see in the literature how much rutin is useful in the drug resistance epilepsy, and you have added a Boolean operator or, so what will happen? Your search items will pick up all the articles which have mentions either rutin or DRE. So obviously you will get lot of number of articles. But what is your objective? You want to see in the literature what is the role of rutin in the drug resistance epilepsy. So in that case, what you have to use? Or and? You have to use and, right? So you can see when you are using the proper search items, rutin and DRE. So when you are using the and, so the literature which has mentions rutin as well as DRE will only picked up from the database. If you are using or, then it will pick all. जहाँ जहाँ rutin होगा वो सारे article वो आपको दे देगा. जहाँ जहाँ DRE होगा सब दे देगा. So you will get lot of article, maybe 1 lakh, 2 lakh, 3 lakhs articles. So it's very difficult to screen these articles. So best way to how to do the data mining, you have to understand how to use these search items. There are a mass builder as well nowadays, AI based. So it will restrict the few. So it will pick up only those articles which are applicable as per your objective. Next slide, please. So like PubMed, you have to go through the advanced. Don't put here in the search window. You just click on the advanced. Next slide. So in the advanced, you have to be click and then you have to be make this search item. So you can see when you have added rutin or drug resistance epilepsy, how many articles? 19,937 results. Now go to the next slide. And when you have added rutin and drug resistance epilepsy, so how many you got? 499, whatever it is. So how much number has been reduced? And which one is applicable according to your objective? You want to see, you are not want to see role of rutin in anywhere. Your research question is very defined. You want to see role of rutin in drug resistance epilepsy only. So you want only that literature. So you have to do that process. Next slide. So like that, then next step you have to be, because again, if you are getting 499 article, practically is it possible nowadays such kind of students will read all these articles 499? No. So next step is you have to screen the studies as per the title. And in students' mind, title should means their aim and objective should be clear. What they are going to check in the literature. So by reading the title, students can identify, okay, this one is important or not as per my aim and objective. So here you can see there is a box. Here you can click and you can select which one are important. Another one you remain the unselect. So automatically your whatever you have not selected, it will be excluded. You will get the number. Next slide, please. So you can select option here abstract. So the article which you have filled important and you have clicked on the tick options, only that article abstract will be generated automatically for you. So that is one of the data mining. Next slide, please. Now we have this Rian as well, which we are doing in our lab as well. This is a AI based tool, and here also you can create your login ID and passwords. It's available commercially as well as freely available. So by Rian tool, your literature process you can do the faster way. So in that one you can do blinding as well. If you have assigned literature to do on this topic to two students, to both students can be blinded to each other by this tool. And guide can see whatever you have done so far. And you can put comment as well. So this is the beauty of this AI based SLR, right? Next slide, please. It's very user friendly to use, but you can track each and everything. Then next step is identification of common target. Next slide, please. That is a step 3. As I told you, Vani. Vani is the tool which will provide you the common target. So one side you have to edit the target which your compound can hit. And another box you have to edit target which are involved in disease. So it will give you the common target. Next slide, please. So like that. So in this case, you can see these are the drug target and these are the disease target. So in the result, you can see automatically you will get the common target between the compounds and between the disease. So you just click on, there is 35 targets which are common between the drug and between the disease. Next slide, please. The next step is interactions among the targets which you have identified. Next slide. So as I said, there is a string. Just type in a Google string database. This is a functional protein association network. Next slide. So this is a string established by European Biology Laboratory in 2019, and it's very powerful tool. Many research studies are using this tool string to check the interactions among the target, so that you can put the cutoff values as well. If you are playing with the cutoff values, so your type 1 error or type 2, if you are controlling the type 1 error, your type 2 error will increase. Type 2 error represent your power of study, right? Type 1 error is representing your p value. It's rejecting null hypothesis when actually it's true. That is why your type 1 error. And inverse of it is your type 2 error. So that we have to do, and we have to see what cutoff values we have to do for the particular projects. Next slide, please. So this is a string web page. This is a protein-protein interaction network. Next slide. So here you have to add these common targets. Again and again I am telling, these software को use करना, it's not a big deal. To use the software in a proper way, that's very important. Next slide, please. So you have to just type the copy the common target which you have identified from the Venn tool in the string. Next slide, please. And you select the species, Homo sapien, musculus, whatever you are planning to do experimentally. You select it. Next slide, please. And you will get such kind of result. But you are not getting anything. It's good for publications, or you can put in the supplementary data. But now the network analysis will become more important. What are the nodes? What are the edges? What are the shortest path? What are the betweenness? Next slide, please. These you have to do next. I have added screenshot each and everything. There are some manual work you also have to do. Like these files you will got, so how to convert into the Excel file. Next slides. So these kind of Excel works, we have to do some manual work. I have added a screenshot of each and every step. If anyone of you want to do or comfortable with the Excel, so you can do it, segregations of data, and you can identify according to your knowledge what are the signaling pathways which are involved in that particular disease. Next slide. So the BioGrade is another tool by which we are checking the interaction of proteins. Next slide. I think time is already over. So this is the BioGrade. Next. Hello. This is the DIP. DIP is another one. Next. This is the web page of DIP. Next slide. This is the INTACT. INTACT is a freely open source, currently supported by European Molecular Biology Laboratory core funding. Next slide. And this is the web page of INTACT Molecular Interaction Database. Next slide. And this one also you have to do some VLOOK work, VLOOK formulas you have to apply and segregate the targets. Next slide. The next step is you have to create the network. Next. So for the network there is a cytoscape. Next. So this is just type in a Google cytoscape, you will find it. Next. You can download it. It's not the latest versions, but you can install in your laptop. Next. So in cytoscape, these kind of steps you have to do. You have to upload the files and you can create the network. Next. So like that you can create and you can present network in a different ways. Next. So like that we have to do, and also there is a tool by which you have to analyze the network, and in analysis you will get the values. How many nodes are there? How many edges? How many hubs? What is the betweenness? Which one is the shortest path? Next slide. So CAG is so will tell you about what are the pathways which are involved. Next. So this is the CAG, string CAG, where you can get the gene enrichment analysis. You can do it. Next. So like that there is enrichment bubbles which you can do by this one, and you can get the pathways information. Next. So like that you can see these are the pathways which are involved. PI3, AKT, pathway in cancer, and like that node-like receptor, apoptosis. Next. This is another tool, CynoGeo, by which you can also identify the pathway. Next. Then interactions of drug among new chemical. After the network, After this, you will find in the research studies they are coupling it with the molecular docking, and many of you are doing molecular docking. Molecular docking is such a nowadays everyone is doing. So you can couple, because network pharmacology is telling you about the possible target, but how your compound is interacting with the possible target you can get by the molecular docking. Means molecular docking is a method by which you can predict energetically favored binding conformation of the ligand in the active site of the target, right? That is the molecular docking. Next step. So you have to couple it with the next, with the molecular docking. So I am not going in detail, but we are doing with the Schrödingers in our lab these molecular docking. Next slides. Then there are how to select the target, what are the different ways. Next ligand preparations. Next many of target preparations. Next grid generations. Next then you can do site map analysis, particularly for the protein in which internal ligand is not available. Next then you can do the docking, and particularly flexible docking. Next बस खत्म कर रहा हूँ. MMGBS calculations. Next आपके हाथ में ही है रिमोट, आप फटाफट कर लो. Next, so you have to couple molecular docking with the molecular dynamics. अगर आपको complete set of studies you want to do network pharmacology, molecular docking, and then do the molecular dynamics with top hit compounds. So molecular dynamics we are doing, and it's depend upon Newton's second law, F is equal to ma. Next. The molecular dynamics also you can do. Next. Yeah, so these are the parameters. I think many of you knows in molecular dynamics we are calculating RMSD, RMSF, radius of gyration, potential energies, SASA, all these parameters, PCA, free energy landscape. So all these parameters we are doing to check the stability of the compound in the active site of the targets which we have identified. Next, these are the PCA. Popularity of network. Next. So you can see in the literatures how many studies, research article has been published in the last 10 years, 20 years with respect to network pharmacology. Network pharmacology is people are integrating with the experimental approaches. Now you plan for animal experimental studies. Just do 1 or 2 essays, you can correlate whatever the targets you get, and you can hit the important literature. Next. So why not India? Because we have a herbals, lot of herbals we have. As I said, China is doing extensively network pharmacology on traditional Chinese medicines. And if you go through in India, how many researchers are using network pharmacology, you will find very few of them are doing. So why not us? And why not? And major opportunity with network pharmacology, you are not dependent on chemicals. It's up to you how much you can explore it, right? So Indians are in top 10% scientists. Indians are very good in programming or computational things. So I think we have to use these network pharmacologies with respect to our herbal medicines. Next. So recent work which we have done in our lab in DIPSA. Next. So this one we have identified some compounds with respect to drug resistance epilepsy. Next. And these are our major findings. We have combined it with the experimental approaches. We have done one animal experimental studies after that. And then we have correlated these computational findings with the experimental studies. Next. These are another studies which we have explored the selected phytoconstituent of galactose and combined with the experimental approaches as well. Next, animal experimental studies. This is another studies which we have done Tinospora cordifolia with JEV vaccine. So we have done network and then experimental. Next. This is another one. Next, with respect to adverse drug. Next, calcitonin. These are the publications which we have done recently. Next. These are the one of the journal which I am the associate editor, and I invite all of you, if you have any paper with respect to network pharmacology in traditional medicines, you can submit it. Next. This is another journals where I am associate editors, and we have open call for the role of network pharmacology integrated with experimental. Next. Challenges: insufficient data, quality, multiple data sources. Next. खत्म नहीं हो रहा है. Conclusion. Next. So I would like to acknowledge my funding agencies, DST Serve, where I got 2 projects, DST SRG, CRG, ICMR, 4 projects running, Sun Pharma, where I have worked on signal analysis, IIT Delhi, where I have worked as a postdoc fellows in the supercomputer facilities, Naipa Library, where I worked as, BITS Misra, where I done my PhD, THIST, this is my first job as a research scientist, then ISF Moga as a associate professor and my current organization. Next. Thank you so much. So thank you so much for your patience.
Thank you so much, sir, for such a nice and knowledgeable session. Any questions? Now, I would like to request our session chair, Radha ma'am, to concluding remarks.
Good evening, everyone. Thanks, Dr. Anup, for such an informative lecture on informative network pharmacology for the study of herbal medicines that forms a bridging the gap between your traditional knowledge and modern medicine. Basically, its application in drug discovery and personalized medicines, which could transform how we understand and utilize the herbal medicines, which could be a benefit for our future. Thank you, Dr. Anup.
Thank you so much, ma'am. Now, I would like to request Radha ma'am to felicitate our speaker with momentum. A big round of applause. Thanks, ma'am and sir. Now, I would like to request Dr. Nitin Kumar, Professor, Department of Pharmaceutical Technology, MIT, Meerut, to felicitate the session chairperson with memento and a certificate. A big round of applause.
Thank you so much, Dr. Mansi. Now, I would like to request another session chairs to call upon, and I would like Dr. Radha ma'am and Dr. Anup sir to take the chairs, please. Thank you.
Here.
I would like to call upon Dr. Lubhan. We'll wait for a 2 minutes gap. Now I would like to request Dr. Anup Kumar sir and Dr. Radha Goyal for being the session chair for the next session. Please sir, I kindly invite you for the next session. Please give a big round of applause. People are less here, please show some energy. Thank you for the motivation. Thank you, ma'am. Thank you, sir. I will request Dr. Lubhan, sir, who is working as a professor and head of the Department of Pharmacology in Subharti University. He's having more than 15 years of experience as an academician and a researcher. Thank you, Dr. Lubhan. Now I would like to invite our speaker for the day, Dr. Himanshu, who is Head Regulatory Essential Roots, New Delhi, to join on the stage, please. Dr. Himanshu. Prem Shankar Misra, sir, to join for the session chair. Mr. Prem Shankar Misra is currently working as an associate professor with the Department of Pharmacy, School of Medical and Allied Sciences, Galgotias University, Greater Noida. He has worked as an assistant professor in the Department of Pharmacy, Meerut Institute of Engineering and Technology, Meerut Sai, in 2010 to 2017. Thank you so much, sir. Now I request Dr. Avinash Singh, sir. HOD Biotechnology, MIET, to felicitate the speaker with a bouquet. Now I request Dr. Avinash Singh, HOD Department of Biotechnology, MIET, to felicitate our speaker, Dr. Himanshu, with a bouquet. Thank you so much, sir. I would request Dr. Avinash Singh, sir, HOD Department of Biotechnology, Meerut, to felicitate our session chairpersons also with the bouquet. Dr. Prem Shankar Misra, and Dr. Lubhan Singh. Please give a big round of applause. Come closer. Come closer. That will be working. Come closer. Thank you, sir. Now, we do have a high tea after this session. So, keep your energy still on the high notes so that we can enjoy our next, a very interesting session as well. Thank you so much for that. Now, I would like to invite our session chairpersons, Dr. Luban Singh, for the introduction of our speaker, Dr. Himanshu, who is Head Regulatory Essential Routes, New Delhi. Please, sir.
Thank you very much, ma'am. First of all, I would like to thank to the organizing secretary and the management of the MIET. Basically, it's my privilege to introduce Dr. Himanshu Sharma. So for this session, basically the speaker for this session. So Dr. Himanshu Sharma is a seasoned professional with over 15 years of experience in regulatory affairs, clinical research and scientific writing. He is currently serving as a regulatory and scientific advisor at Essential Routes Provided Limited since April 2024. Sir. In this role, he collaborates with government authorities such as FSSAI, CDSCO, that is the SFDA, and the insecticide boards, and managing regulatory compliance and registration for medical devices, food, supplements, OTC, FMCG, and pharmaceutical products. He also develops basically the scientific content for global regulatory submissions and oversees clinical trial monitoring and project management. And previously, Dr. Himanshu Sharma was the Senior Principal Scientist, Clinical and Regulatory Affairs at Patanjali Research Foundation Trust up to June 2023 to April 2024, where he led a team of the 18 CRH managed clinical studies and led the regulatory submissions for Patanjali products. He has also worked as a freelance regulatory scientist up to the 2020 to 23, collaborating with various government bodies and overseeing compliance for a wide range of products. Dr. Himanshu Sharma has held senior roles in regulatory affairs at the Glenvia Performance Nutrition, Amway India, where he managed the compliance and registration for food and supplements and lies with the FSSI and other regulatory bodies. His earlier career includes senior manager positions at Sun Pharmaceuticals Industries Limited, where he contributed to regulatory strategy basically and evaluated the product proposals and managed the clinical trials in line with the ICH GCP guidelines. He holds a PhD in pharmaceutical medicine from Jamia Ahmedabad, along with an M.Pharm in pharmacy practice and bachelor's in pharmacy. Additionally, he is certified in food safety management, ISO 20018, advertising code by SCI, US FDA, IND filings and OTC products, so many things he have gained during this tenure. And his extensive experience spans regulatory strategy, clinical trials, scientific writing and policy advocacy, making him. Sir, thank you very much for just giving the consent, and really I would like to thank the organizing committee again just to introduce such type of person and the personality. Thank you very much, sir. So now over to you, please. We are eagerly waiting for your session, sir. Thank you very much. मुझे उधर देखना पड़ेगा बाद में.
First of all, thank you, audience, for being patient. आप पास में भी आ सकते हैं, because I cannot fit इतनी दूर तक जाएगा नहीं, and आप आराम से आ जाइए, आप पास में आ जाइए. Please whenever try लो. आ जाइए, आगे आ जाओ, कोई आराम आ जाइए.
सामान छोड़ दो.
Sometimes English is difficult, so I will use English. I will mix English and make the things lighter for myself, not for you. मेरे लिए आसान हो जाएगा, आपके लिए difficult हो सकता है. आगे आ जाओ, आगे आ जाइए. क्या दिक्कत है? Because in a post election से जो रजिस्टर सेशन होता है और वो भी आपका की वेट कर रहा है, तो मेरे लिए काफ़ी डिफिकल्टी है. तो मैं आपको कैसे एंगेज करूँ? But the opportunity has provided me to engage you with the understanding on the regulatory landscape for the health and the herbal medicine. So if we look what is the regulatory landscape, so regulatory landscape is nothing but looking how the things can be reached into the market in legitimate way. When we talk about the herbal medicine and the botanicals, we can broadly classify them in two categories. So first is a prescription, the other one is a your consent choice, like you can say over-the-counter product, supplement, or the natural health product, where you don't require any assistance from the prescriber to select a product. You can identify your condition, you can select a product, you can then decide whether you want to take it and how long you want to take it. That is called a self-prescription, self-medication product, which are quite safe when you take continuously, or you can take them as per your will. Nobody force you, nobody prescribe you, nobody induce you. The other type of products are those which are medicine, which require it's like a condition, you require a driver to run the vehicle. So doctor will advise you. He may be an MBBS doctor, so it become a prescription medicine. If he is your means BMS doctor or something, and means traditional medicine practitioner, then it become a complement in the traditional medicine. So this is a broad category. If you look who control this medicine, so it is not like a means globally recognized by a single authority. Every country has their own territory, and the authority within the country are different. Like in case of India, it is two authority which controls the herbal and the botanical product. Similarly, in the US, Canada, and the other places, the authorities are very much different. So we will have a flavor of each of main regulatory body and look what is the requirement. So we are in India. So first of all, we should understand what is the requirement in India. So herbal and botanical products are classified first of all as a medicine. So these are controlled by as a drug. So I will be touching on the same. Then my slide will be on the positioning the botanical as a supplement and where these are regulated as a supplement. I will be touching those requirements, and then finally as a food where they are endorsed that these are considered as a food. So in my next slide, you will find that within the Drug and Cosmetic Act 1940, the regulation has clearly defined that what are the Ayurveda, Siddha, Unani and Tibetan medicine, and where these are documented that these are the medicine which can be used, which are the traditional use, that is these are the safe for centuries and have been used and can be used as per the recommendation of a prescriber or the self-medication. So these are the various regulations, rules are provided and schedules are provided. Like I am touching on the key aspect. Like in the first schedule, we will find a list of authoritative textbook. Like these are the books from where if you want to take a license, because we here are learning for the opening either of our pharmacies or means working somewhere in a pharmaceutical industry. So we should know that which are the product or which are the ingredients which are positive, which can be referred in this book and then can be taken thereafter to the regulator. Then there is a second schedule which provides that what are the standard. So if we prepare a product, it is not like we just pick neem from the garden and take it saying that it is becoming your medicine as such. It is not like that. There is some standardization is required. So if we are taking those things, then certain standard to be followed, it is mentioned within the schedule 2. And then we are talking about what are the different formulation, different type of formulation which are permissible. So as we know that for most of the Ayurveda product, oral and the topical products are allowed. And then finally, what are the list of various poisonous substances which can be poison. These are also written in the rule book. And not least, but the good manufacturing requirement is mentioned under the Schedule T. So these are some of the things which we learn as we practice those things. It is not, we can just inject you in a daily dose like this. So now talking about the herbal product as a supplement or as a nutraceutical. So these are well documented into the nutraceutical
regulations which introduced in implemented in 2018, which has more than your 1,000 odd products, which includes more than 400 to 500 and a herbal products which can be used as a supplement. So if we look these things, these are becoming a more easier to understand. तो मुझे लगता है कि मुझे थोड़ा सा जल्दी से करना पड़ेगा इस चीज़ को, क्योंकि आपकी बस का भी टाइम हो रहा होगा। तो यहाँ पर एक लिस्ट दिया हुआ है और लिस्ट में से आप सिलेक्ट करके प्रोडक्ट को ले सकते हैं और उस प्रोडक्ट के अकॉर्डिंग कंपाइल करके रिक्वायरमेंट को फुलफिल करके आप एक सप्लीमेंट बना सकते हैं। व्हेन इट कम्स इन अ टैबलेट कैप्सूल डोजेस फॉर्म, देन इट बिकम अ योर सप्लीमेंट। इन द रेगुलेशन, न्यूट्रा रेगुलेशन के बाद में एक नया रेगुलेशन आया है, उसी के अंदर आयुर्वेद आहार। सो व्हाई इट इज इंट्रोड्यूस्ड एंड व्हाट इज इट इज इम्पॉर्टेंट, बिकॉज आवर टॉपिक वाज regulatory pathway for botanical and the botanical product or the herbal product. So as a food, these products are approved within the drug, within the Nutra. But why there is a need to have a Nutra Ayurveda Ahar? Only because the food like Chamanprash and other things, these are registered in other countries as a food. In India, these are considered as a Ayurveda product. So that's why there is a need to take such product to give a global recognition. In the country of origin, it is also as a food, and now in the free sale certificate, if we have to export the things or import the things, then the free sale certificate become an important part, and that's why this landscape change will provide help to the industry. In European Union, there is a simple classification of the herbal product. They consider as a traditional use registration. Registration is different from license. Like somebody put a registration, it is, you can take it as a requirement or lay down, and somebody has to self-declare it. So if a product contain a ingredient or any ingredient is used for more than 30 years without any significant safety and efficacy results, so it will be considered as under the traditional use, and a company can provide the data and take a registration under that. Whereas the well-established use, the requirement is little less. It is a 15 years of use, but here the organization or the authority has to first scrutinize the application and then provide you the approval. And same stand for your standalone and the mixed application, where the ingredients, if it is isolated from the herbal product, has to be treated more robustly, and the clinical trial may be required in that case. WHO has also provided the guidance for the regulation on the herbal medicine in the Southeast Asia. So why it is specific to Southeast Asia? Because Chinese medicines are different, and in South Africa they have their own traditional medicine. So that's why this has become important to test that whether the Indian regulations which are formed, they are in line with the WHO requirement or not. So WHO has classified again the herbal product or the medicine as under the 5 categories. Here it is written only 4. So they are the category where indigenous herbs are there, then herbs medicine in the system, and the modified herbal medicine, and the imported product with the herbal medicine. So these are mentioned within the guidance document. So moving ahead, there is a certain minimum requirement. What should be a requirement for the safety and the efficacy? These are also listed within the same guidance document, where the requirement of toxicity study, as we are discussing that some of the toxicity study has been conducted by the institution, and Dr. Agrawal is involved with that. So I was discussing that how they are conducting, whether their labs are equipped with some type of facility. So she assured that she is fulfilling all the legal requirement to conduct such type of study, and we are grateful to know more about the institution. Then coming to what are basically the efficacy data which is required for a different type of a disease condition, because certain conditions are acute. If I have to take a medicine for the headache, I cannot look at the long-term study for the safety as well as the efficacy. So considering the requirement, my requirement for toxicity as well as the efficacy changes. So clinical studies might be required in the acute cases, and similarly for the chronic cases, preclinical study may be relaxed, considering that the herbs which is used whether it has a traditional use in the market or not. If it is used for centuries like neem, if we are want to repurpose it as an antipyretic, so people will have a less difficulty in accepting the safety because it is well established. People are consuming neem, people are consuming tulsi and other products as well. So requirement for the preclinical study may be waived off or they can be abridged. Then comes the health condition. So if a product is for the little element, then the requirement for the preclinical and the clinical study become a little narrow. We can basically look at the requirement and then move ahead without going into the lengthy requirement of a clinical development. So similarly, the requirement for the dosage form, mode of administration, these all are the requirement which has to be disclosed when we obtain the license for the herbal product. So sometime when the product which we wish to launch in the market, it has a activity mainly into the leaves, and some other products have a roots. If somebody want to use like a ashwagandha leaves, but whereas we know that ashwagandha is mainly used for roots, then the person has to done certain studies to prove that. And people are now using the ashwagandha leaves as well, but they have a data to prove their efficacy and they repurpose the product in that category. So depending upon the requirement and depending upon the dialogue with the regulator, we provide a justification and then plan what is the actual requirement. So everything is okay. We require certain clinical trial, we require certain preclinical study, but without quality there is nothing which will be moving, because if we are not consistent, we do not validate the data, which we do not have any validation, what is the batch to batch, or you can say raw material are same or they are different in terms of their efficacy and the potency. So that's why there is a requirement that there must be a regulatory authority within the country or the territory. Import license is required if the product has to be imported. Like suppose if cinnamon we are importing from Sri Lanka, and cinnamon which we are procuring from southern part. So the efficacy of the key component or the, you can say, some of the ingredient may not be safe. That's why it is required that an authority will evaluate their import, and the product information must be submitted during the time of registration. And what should be the minimum criteria that product information must be there. So I will be taking you through in the next slide. And there should be a guideline for the good agriculture practices and the collection practices. If somebody has want to collect a product in autumn season, whereas the efficacy is more in winter season, then we will not find the marker compound and we will fail to achieve a consistency. So that's why it is important to define the good agriculture and the collection practices. We should have a practice to do a quality control, and not the least, the product which is being manufactured, it must be manufactured in the good manufacturing facility. It is not like that we know that घर पे दादी जो है कढ़ाई पे बनाती थी तो च्यवनप्राश में भी सारा कुछ लेके ऐसे डाल देंगे, उसको मार्केट में हम लेके आ जाएंगे, तो वो नहीं हो सकता. We have to follow the good manufacturing practice. If we are marketing the product, if we are manufacturing for own, so we are risking only ourselves. So it is out of the purview of a marketing. So in the product information requirement, it is the same as for the general your pharmaceutical product. The part which are important is we must provide the information related to the specific part of the herbs which we are taking, the quantity, and if there is a possibility to have qualitative and quantitative analysis, it is also required here. Pharmacovigilance is important. I have seen various cases when I was doing my ward round during the MPharm, that many of the products which are taking herbal products, get a diarrhea or edema, and that's why they lose their interest in the long term therapy. So उसको देख के ऐसा लगता है कि फार्माकोविजिलेंस की एक इम्पोर्टेंट रिक्वायरमेंट है और जो होना चाहिए. So I am moving to the last part. In India, the requirements for conduct of a clinical trial or the drug development are well defined within the regulation. और इन रेगुलेशन को देख के आप खुद भी इसको कर सकते हैं. कंसिडरिंग आपका टाइम हो रहा है, आपकी बसेस होंगी, मैं यहाँ पे इसको डिटेल में नहीं जाता. आई विल गो फॉर डायरेक्टली फॉर द 6. So आपको ये सब मिल जाएगा इस गाइडलाइन के अंदर, तो मुझे रिपीट करने की जरूरत नहीं है. Then comes the phytopharmaceuticals. So these are again you can say from the subset of the herb product. So in the phytopharmaceutics, it is now well recognized within the new NDCT rule, new clinical trial rule, in which the requirement for the quality, conduct of a trial and their application is well defined. So इसको भी यहाँ पे